Hello students, good evening you all. Today we are going to discuss biotechnology application chapter. So far lot of chapters, complicated chapters, high weightage chapters we have been completed. Today I am with you I am here with you a particular chapter, biotechnology application. All of you know biotechnology application and biotechnology process, two chapters contribute 12 marks. The weightage of this chapter, that means this unit, 12 marks. So we have been already completed one important chapter called biotechnology process chapter. Today applications of biotechnology chapter. So I hope these all classes are helping for you people to brush up your concept. Today I am going to discuss few important questions from this chapter. Moreover student, today's questions are statement based questions. So today majority of questions are statement based question, 2 mark question, 3 mark questions also there. So kindly all of you keep NCERT also with you. I will show the reference from the NCERT. If possible, all of you open the NCERT and keep it with you. Am I clear? So shall we start with the first question? Look at the first question in biotechnology application chapter. The first question in reason and assertion based question. If you know the answer of this question, kindly inbox your answer. Look at the assertion first. What assertion is given to us? The assertion is the ADA enzyme, ADA gene provide instruction for the producing enzyme adenosine deaminase. Is this statement correct or not? ADA gene provide the instruction to give adenosine deaminase enzyme. The statement is correct. Am I clear? The statement is correct. This gene undergo transcription translation to give a specific enzyme, the enzyme called adenosine deaminase enzyme. This enzyme is essential for the maturation of B, T, both cells, B and T lymphocyte cells. Done. This statement is correct. What is the reason is given here? This enzyme is found throughout the body, but this must activate in lymphocyte cells is given. Is this statement correct? Is this statement correct? Yes, this is exactly correct statement. What is the statement? The whole body, this is a gene present in whole body, but exclusively it functional in where? T cells and B cells. But what is the right answer? Is it, shall I go for the A or B? What is the very good Hafiz, Ganga, Ritika, absolutely correct. So the right answer I can go. Both statements are correct, but the B statement, that the reason is not correct explanation for the assertion. So I am going with what? Option B. Am I correct? Am I clear till here? Everything clear? Adenosine deaminase enzyme gene therapy question. Lot of questions are today we are going to discuss. This is one of the important topic for your 5 mark question. 100% ADA gene therapy questions are going to come. So all of you kindly focus on the board. Lot of different questions are going to come on the gene therapy question. So first question, option B. Second question also assertion and reason based question. Second question also assertion reason. Look at the assertion. What the assertion tell you? RNAi is a cellular defense mechanism in eukaryotes. RNAi, that is RNA interference mechanism, is a cellular defense mechanism in all eukaryotic. Statement correct or not? I need statement correct or not? Yes sir, statement is correct. Absolutely, statement correct. This is the thing. This is the RNAi mechanism we are using to rid of that what? Nematoda. What is the nematoda? Melanogain incognition nematoda in tobacco plants. It's correct. Now what is the reason here? It is silencing the specific tRNA. It is silencing the specific tRNA or specific mRNA. It is silencing specific mRNA, right? So if you are able to recall the molecular basis of inheritance, what was given there? What was given? Ritika, right? Good. Allah, right? Sana, what was given in the molecular basis of inheritance? The gene expression or the regulation of gene in eukaryotic organism can stop or can regulate in 4 levels. It could maybe transcription, translation, processing level or splicing level. Here eukaryotic RNAi mechanism shutting down the gene or silencing the gene at translation level. Transcription, mRNA is forming, but this mRNA is not allowing to go to what? Tell me, translation. So the essential protein for this nematodal growth is not going to form. So nematodal growth is not takes place. Remember all of you, this is not tRNA, it is mRNA. So I can go what? Tell me, assertion is true but reason is false. So option C. First two question, assertion reason based question. Here after all statement based questions are coming. So everyone focus on the board and try to read all the statement, which one is correct, which one is incorrect, try to answer me. Look at here, the third question. Third question is, which of the following statement are correct about the adenosine deaminase deficiency? I can simply call SCID, right? Severe combined immunodeficiency. This is a syndrome, it's a problem, this is a gene problem, it's a gene mutation, right? So they are asking what? In the absence of adenosine deaminase enzyme, purine metabolism disrupt. Did I say this one? Purine metabolism, NCRT is there in the purine metabolism, this one? No sir. Because of ADA gene deficiency or ADA enzyme deficiency, immune power, acquired immune system, immune power is getting down, right? So this statement is not correct. So I am keeping it is not correct. Now ADA deficiency is caused by the deletion of a gene for the ADA. Is this statement correct? I am not getting answer. Good, right, very good. Ganga, what is the right answer of this? The second statement is right or wrong? Deletion or addition, or it is a point mutation? What is this? Yes sir, it is a deletion, right? It is a deletion. The gene is completely deleting. There is no gene for what? A code for the adenosine deaminase enzyme. So it is a deletion. Fine. So this statement is correct. Fine. Good. Now option 3. The ADA patients can be permanently cured by bone marrow transplantation and enzyme replacement therapy. NCRT given there are 3 solutions are there to cure this disease. One is called bone marrow transplantation and one is called enzyme replacement therapy. Third one is called gene therapy. But which one is permanently cure? Gene therapy, and that to where? Early embryonic stage, right? So this statement is also wrong. So this statement also wrong. So I am going the last one. For permanent cure, genes isolated from bone marrow, correct. Cells producing ADA, introducing into the cell early embryonic stage, absolutely. Early embryonic stage. So I can write the right answer is right. Good, very good. So first one is wrong, second one and also fourth one. So option B, very good. ADA enzyme therapy permanently we can cure if you do this gene therapy or introducing this gene into the early embryonic stage. We can permanently cure this one, right? Done. Next question number 4. Question number 4. Any one of you? Any one of you? Shadi, Sana, Ritika. Next Samuel. Which statement is correct about the golden rice? Golden rice I'm talking about here. Biofortification, increasing the vitamin content, increasing the protein content in a crops. It is a golden rice, vitamin A enriched crop. So vitamin A enriched crop here, it is a transgenic variety of rice. Okay, fine. It contains good quality of beta carotene, pro vitamin A, absolutely correct. Beta carotene is principal source of vitamin C. Is this correct? Vitamin C? No sir, it is not vitamin A. Now the grains of the rice are red in color. Red in color due to the beta carotene. Is this red in color? No, right? So the right answer is any one of you? Any one of you? The first 2 statements are correct. So I can go 1 and 2. Absolutely. This is a transgenic crop. Transgenic crop enriched of vitamin A. Biofortification also you should remember. Increase the protein content or vitamin content in the crops. This we can say it's a biofortification process. Am I clear? Golden rice is one of the transgenic rice which is enriched of vitamin A to avoid or to prevent night blindness and also what this eye problems. Am I clear? Next question. Next question. Find the correct statement from the following. They are asking correct statement from the following. All 4 statements are given here. I will read one by one. I hope you will give the right answer. The gene therapy is a genetic engineering technique used to treat disease at molecular level. Tell me, option 1. First option is correct or wrong? It is a molecular level we can treat or not? molecular level wise we can treat or not. Yes sir, we can write what may gene therapy is a genetic engineering. Genetic engineering is a technique, rDNA technique to cure the diseases in gene level or molecular basis level. Correct. Now I am writing here, it is correct. Calcitonin is medicinally useful for the recombinant produced in the treatment of infertility. What is calcitonin? Do you remember the C cells of thyroid gland secrete calcitonin? Any one of you can tell me the function of calcitonin? Function of calcitonin. This statement is not correct, but tell me what is the function of this calcitonin? Increase the calcitonin will participate in the calcium metabolism in the blood. Am I clear? It reduces the calcium level in the blood. It takes the calcium from the blood, extra amount of calcium, it deposits in the bones, and here the calcium metabolism. Fine. Now it is not in where? Infertility. It is a wrong statement. Leave it. Next, Bt toxin is biodegradable insecticide obtained from the Bacillus thuringiensis. We know this one. Bt toxin is a what they call bio insecticidal protein coming from where? Obtaining from Bacillus thuringiensis bacteria. Cry gene, cry protein, we have already discussed. This statement also correct. So I can go with 193. Where is 193? 193 is the right option. Answer C. Right, very good, absolutely. I will be silent for the next question. Next question, complete dedicating to you. Whoever attending online, you have to read, you have to answer. Okay, look at the next question. Question number 6. This is a little big one. I will read and help you. Chalo, pick up right answer. Okay, I will not help you to identify the answer. Pick up the right answer. Consider the following statements are given. Earlier, insulin was extracted from pancreas of sludged cattle and pigs, which was more efficient than the genetically engineering insulin. Is this statement right or wrong? This is a more efficient than the rDNA technology insulin. Is this right or wrong? Sir, wait a moment. How come if it is a more efficient, why would we go for the rDNA technology? It is no, it's wrong. It's completely false. Because of this one, the patients are showing what? Allergic reactions. When they're showing the allergic reaction, so biotechnology is decided. What is that? We have a pathway to get that good human insulin with from the microorganism. So that's called what? Through rDNA technology. So this statement is wrong. So this statement is wrong. Good. PCR technique is being used for the detection of HIV in suspected AIDS patients and genetic mutation in suspected cancer patient. 1 minute, everyone. Look at here, everyone. PCR. If you have NCERT in your hand, I will ask you now, everyone, open the NCERT of molecular diagnosis topic. In the molecular diagnosis topic, early detection of diseases is possible by 3 ways is given. One is called rDNA technology, PCR, ELISA. rDNA technology, PCR and ELISA, early detection of diseases. You go to doctor when you have a lot of symptoms. Student, yes, Hafiz, Shadi, when we go to doctor, we have a lot of symptoms of vomiting or whatever this, a fever or this, all the symptoms. That mean already in our body, the bacteria, the pathogen content is concentration maximum. So what doctor will identify based on these symptoms, they will give for the urine test or blood test or something. But the patient don't have any symptoms. It's good only early detection of diseases, no symptoms, nothing. But we can identify the disease is possible with PCR. Today we will discuss RT-PCR, next real-time PCR difference also. How can we identify this early detection of what they call RNA or DNA with RT-PCR? Now this statement is absolutely correct. Mutant gene or suspected cancer gene also we can identify. This is the early detection of diseases process. You got my point? Now, next, cystic fibrosis, hemophilia, skin diseases gene treated by the gene therapy. Third option correct or wrong? I need third option, option 3. That means third statement, I'm sorry, third statement is correct or not? Any one of you? Any one of you? It's correct, sir. Right, done. Very good. So I can go with the answer is what? Tell me. Second one, third one correct. Second one and third one absolutely. So statement based question. Why did I select? You know, statement based question will help you to answer 3 mark question also. Got my point? So statement based question will help you to pick up the complete concept clearly. So which one is correct, which one is wrong? Next question also, carefully read and answer the question. Question number 7. Question number 7. Read the following statements. Again, statement based question. Pigs are the most preferred mammals for study of gene transfer. Is this true? Pigs are more preferable or mice? Which one is the best one? Pig or mice? They go for the mice, right? Not pigs, right? So this is keep aside. Next. A transgene is a foreign gene incorporated of which of the which in host cell can make it transgenic. Is it correct or not? This statement is correct or not? Second one is right or not? The a transgenic gene or foreign gene incorporated in the host cell. Yeah, it is called transgenic animal or GMO, genetically modified organism, transgenic animal. ADA deficiency cause severely combined immunodeficiency, SCID, skin disease, gene mutation, gene deletion, ADA deficiency. True. This one is true. Next, 2 insulin polypeptides are cross-linked via hydrogen bonds or disulfide bonds. Hydrogen, we know it is a disulfide but not hydrogen. You have to be very careful this one. In the exam, they can clearly, all questions, all options you're feeling it's correct. Hydrogen, but no. Look at here. We have already discussed this is a polypeptide chain and it is a B polypeptide chain. B polypeptide chains are bound by what? Tell me. They cross by disulfide bond. So remember all of you, this is not a correct statement. So I'm going, this is wrong statement. So how many are correct? Tell me now. How many are correct? So I can, shall I go for the 1, 2, 3 or 2 and 3, right? So where is the 2 and 3? 2 and 3, option B. Am I correct? Till here everything clear? Absolutely. Disulfide bond. Disulfide bond we have also seen in dash. Except insulin, disulfide bond formation we have also seen in the some places. Anyone? Some particular places we have seen. Sania, where? Can you guess? Ganga, Ganesh, Allah, Sania, Hadia. Sir, disulfide bond we have also seen IG. Where is IG? Right, exactly. Antibody, immunoglobulin, heavy chain, light chain, variable, constant, that we have discussed in that one also, the disulfide bond formation. Absolutely. Question number 8. Question number 8, very important question. Very important question. Whoever don't know the type of cry genes cannot answer this one, I am sure. Tell me how many type of cry genes are there? We know the cry genes are, tell me, one is called cry 1AC, next one is called cry 2AB, next one is called cry 1AB, 3 are there, right? So cry 2AB is cotton bollworm. Is it correct or not? Cry 2AB is sensitive to it. It's very what they call cry 2AB protein will act on where? Called cotton bollworm, right? So cry 2AB and cry 1AB produce toxin that controls. I can go with the corn bore respectively. So I can also write cry 2AB and cry 1AC. These two are, I can write what? Tell me, cotton bollworm. Cotton bollworms. Am I clear? This is cry 1AB. Is it true or not? How many of you correct? How many of you correct? This statement, how many of you correct? Cry gene, application chapter, biotechnology application chapter, cry protein, bacillus thuringiensis, toxic protein genes, toxic protein attack on that what they call different type of genes. Everything you should perfect, my dear students. So there are 3 genes are there. What are they? Cry 1AC, cry 2AB, cotton bollworm, cry 1AB on corn borer. So answer is option A. Lot of typical questions are going to come based on this one. Ready to answer. Is it till here everything clear? Everything clear? Done. Question number 9. Question number 9. Second generation vaccinations are prepared by the recombinant DNA technology. Which of the following are examples of such vaccinations? 1 minute. All of you focus on the board. There are three generation vaccinations are there. First generation vaccination, second generation vaccination, third generation vaccination. Third generation vaccinations are not yet released into the market. They are in the under process. So let me explain first generation and second generation vaccinations. Before I go this first and second generation vaccination, let me give a brief idea about the active immunity. What we discuss in active immunity. Look at here, everyone. This is a particular bacteria or antigen. I'm writing this is antigen. This antigen, when entered in our body immune response, which response, tell me, this is B cells. Am I clear? And also T cells. B cells and T cells are respond. So in our body, B cells will convert into plasma cells and also what, tell me, memory cell. Plasma and also memory cells. Done. Now these plasma cells are providing specific antibodies. Specific antibodies again to specific antigen and effectors. So the memory cells also will be there. This we have seen. Active immunity is that immunity if a live pathogen entered in our body, our B cells will produce that what they call antibody against to this one, but this process is slow and fully response. We already seen. Wait everyone, wait a moment everyone, try to understand first generation vaccination, second generation vaccination. Wait, all of you wait. Now look at here, the first generation vaccination. I'm writing here, it is first generation vaccination. Vaccines, first generation vaccines. That means if you introduce weakened or killed pathogen into the body to create antibodies to activate our immune system, we call the first generation vaccination. For example, polio drops, the oral polio vaccine. Oral polio vaccine is that vaccine is a weakened or attenuated pathogens or killed pathogens are there. Got my point? So that is a simple, what is that, weakened or killed pathogen administration to activate our immune system, we call the first generation vaccination, simple, a polio vaccines. What is the second generation one? In this one, what I said, tell me, this one is killed one, killed or weakened pathogens we can introduce. Done. Example, polio vaccine, oral polio vaccine. Number 2, second generation. Everyone focus on the board. Second generation vaccination by rDNA technology. Read the question carefully. rDNA technology means we have to preparing the particular anti-protein part of the particular organism. Let me take one small example. A particular pathogen is there. That particular pathogen protein is responsible for the disease. So we are taking that the gene which is caused for that protein, that gene introducing into the organism and obtaining that product. That is called what, tell me, rDNA vaccination, second generation vaccinations. Example we can write. What are the examples of cholera vaccination, hepatitis B vaccination? Am I clear? And one more is given, herpes vaccination. Now second generation vaccinations are herpes virus vaccine, hepatitis B virus vaccine, and I can also say cholera. Am I clear? Cholera vaccinations are second generation rDNA vaccination. Everyone note this one. Now, so polio vaccination is a first generation. This is first generation. Am I clear, all of you? Now, our answer will be A and B will come. So remember, first generation vaccination to the difference between first generation, second generation. Done, Mike? Am I clear? Is it clear? Done. Right. Very good. Very good. Next question. Question number 10. Question number 10. Which gene was introduced into the first transgenic cow? What is the name of this cow? What is the name of this cow? If you answer it clearly, read is, I think it is 1997. 97, the first transgenic cow called Rosie. Absolutely, Gala. Very good. Very good. Rosie is a cow. So that Rosie cow, we introduced what? A gene. The gene is human alpha lactalbumin. Anyone can tell me how many grams? What is the volume of what is the amount of this one in 1 liter of milk? What is the Hadia? What is the volume of this one? 2.4 mg, right? Yes or no? 2.4, right? 2.4 mg. Yes. So we can write human alpha lactalbumin. Out of the 4 options, one option is there to treat emphysema like a lung disease. Out of the 4 options, one option is grams, right? Is it gram? Okay, I will change here. It is 2.4 grams. Okay, done. Now out of the 4, one is treating emphysema. Very good, very good. What is that? Alpha 1 antitrypsin. Emphysema like a cigarette smoking, the lung alveoli infection, we can treat this one. So these all are undercomes into biological product. Transgenic animal, biological product, uses of transgenic animals, so biological product. Yes, so this is Rosie, transgenic cow, first transgenic cow. Next one, question number, did I write that answer? Option A, done. Question number 11, very simple question. I will not read, you have to answer me. Albert, Sania, Ganesh, Ritika, Shadi, answer me fast. Simple question. The question is, in India, organization responsible for assessing that safety of introducing genetically modified organisms. A simple GEAC. The GEAC full form, Genetic Engineering Approval Committee. Simple question. I don't want to waste time on this one. I am going to next question. Now, which of the following technique is based on the principal antigen antibody interaction? I am very clear, students. Everyone focus on molecular diagnosis topic. Antigen antibody reaction is the principal based on antigen antibody interaction is what? ELISA. Let me take, I already explained this one more time. Look at here. If you wanted to identify a particular disease is present, particular disease, a pathogen is there or not in a body, we can identify with the enzyme linked immunosorbent assay. Look at here. Here PCR also there, but PCR is not based on the antigen antibody interaction. No. What did I say? Early detection of diseases by 3 methods. Are you following me? Are you there? 3 major ways are there to detect the early diseases. One is called RDNA technology, PCR. Third one is called ELISA. But specifically they given antibody antigen interaction. Listen. A person, for example, this is a person went to a doctor to identify. He is suspecting I have HIV. Okay, you don't have we don't know, even doctor also don't know, there was no symptoms, nothing. So what we are doing, we are taking the serum. Yes, serum. Now in this what they call antigen antibody test, that is a ELISA, there was a micro titrating plates are there like this. Micro titration plates are there. I told you this one. Now look at here. We will administrate a particular suspecting antigen. So this is a suspecting antigen. What is this suspecting antigen? GP120. If you able to recall the human health and disease chapter, I told you the antigen nature of HIV virus is GP120, I said. Right? So the doctor, the diagnostic person wanted to identify whether the HIV is positive or HIV virus is there or not in his person body. What happened? They took the serum and they added here. Okay. Now, if this person already affected by the HIV virus, in his body, antibodies against to this HIV virus must have been there. So when antibodies are there, wherever this GP120 form, there will be a reaction like this. This is antibody and this is antigen. Look at your antigen antibody reaction. So with the help of alkaline phosphatase, the enzyme with the particular color formation, we can identify antigen antibody reaction form. So when antigen antibody reaction form, we can easily tell you already infected with the HIV, because antibodies of HIV we found in your serum. For example, this person did not affected by the HIV. There is no chance of formation of what, tell me, antigen antibody complex formation is not takes place. Unnecessarily our body will not produce antibodies. You got my point? If you infect only our body will produce the antibodies against the antigen. So what is the mechanism? Antigen antibody interaction. So I am going with what? ELISA. Enzyme linked immunosorbent is a alkaline phosphatase we are adding, and we are getting the particular color. When you are exposed to radioactive and this X-rays, a particular color will form. Based on the particular color, antigen antibody reaction is there or not, we can identify. Remember, this type of higher order thinking questions can come. Next question. Which of the following molecule diagnostic technique is used to detect the presence of pathogen in early stage of infection? Early stage of infection, how can we identify? Come on. Any one of you? Karthik, Kaushik Ram, then particular method is given. Read carefully the question options. Angiography, radiography, enzyme replacement technique, and also polymerase chain reaction. What is the right answer? Very good. Yes, you all are following me. Absolutely correct. So PCR, polymerase chain reaction. Early detection of diseases could maybe cancer, HIV, any diseases like this, RNA type of viruses or DNA type of virus. If you are able to recall, there are two type of PCRs are there. Real-time PCR, okay, one PCR we can write real-time PCR. I make clear. In this real-time PCR, we will get DNA to DNA. That means DNA to amplification of DNA. But reverse RT-PCR, you might have well known about this one, RT-PCR test. Did you take the RT-PCR test or not before you go into India? After coming India, we have already ever, right? We have already struggled last 2 years, right? The last 2 years or last 1 year, RT-PCR test. After you get on the airport, after this quarantine, RT-PCR. What is RT-PCR? Look at here. This RT-PCR reverse transcriptase. Now here reverse transcription PCR, that means from mRNA or RNA, from RNA we are getting what? DNA. This is RT-PCR. If you able to recall the last year term 2 examination paper, in the term 2 examination paper this question was there. But what is the full form of RT-PCR? RT-PCR, the real-time PCRs are DNA to DNA formation. But the RT-PCR, this RT means reverse transcription PCR from RNA, that means coronavirus is RNA virus, we are getting that RNA to DNA. Got my point? So this is RT-PCR. So remember all of you, polymerase chain reaction, but we'll learn in our NCERT principle biotechnology process chapter, PCR, amplification of DNA we learn, amplification of gene we learn. Are you following me? Are you there? Right. So the answer is what option? The next question. Everyone know, I think so, or whoever attending the class, everyone know the answer of this question. Shall I get the answer of this question? What triggers the activation of prototoxin? Protoxin activate Bt toxin. It is alkaline pH. Where is the alkaline pH formation? Sir, in the gut of insect. In the mid-gut of insect, alkaline pH form. So immediately the prototoxin can convert into crytoxin. It can make a perforation and ultimately kill what? Insect. We have discussed this one. This is a very simple question. So I can go alkaline pH. Question number 15. I will be silent. I will be silent for a while. Any one of you, online students, be active. All of you, be active. Answer me. Shiba. Next is Shadi, Anisha, Samuel, Hadia, Abin. I need correct answer of this question. Choose the correct option regarding retrovirus. Retrovirus is the what is the example of this retrovirus? HIV virus. Chalo, all of you answer. The right answer is option A. Good, very good. What about others? I need answer. Ritika, good, very good. What about Allah? Right, good. Look at here. The RNA virus that can synthesize DNA during infection. Yes, absolutely. It is a RNA. How many molecule of RNA? How many molecule of RNA? How many molecule of reverse transcriptase enzyme? I need answer. I need answer. You have to tell the answer. How many molecule of RNA? How many molecule of reverse transcriptase? I told you in the last class, absolutely correct. 2 molecule of RNA and 2 molecule of reverse transcriptase enzyme. So this RNA, after injecting into the macrophages and lymphocytes, it convert into cDNA, DNA. We discussed this one. Done. Next question. Very simple question. I'll write something on the board. What is that? Tell me now. Now this is A polypeptide chain. This is B polypeptide chain. Between AB polypeptide chain, C polypeptide chain is there. So let me write here. This is A. This is C. This is B polypeptide chain. So shall I call the mature insulin? Is this mature insulin or pro-insulin? What type of insulin is this? This is a immature or pro-insulin. I can write pro-insulin. Pro-insulin will actually secrete initially, and after that will undergo process to form a mature insulin. What is the answer? C peptide of human insulin, if you are able to recall the NCERT line, it will remove. It will remove when it is convert into what? Mature insulin. So I can go. It responsible for the formation of disulfide bond. And it a part of a mature. No. It is not a part of a mature insulin. Removed during maturation of pro-insulin to insulin. That means mature insulin only have a 2 chains, 2 polypeptide chain. Any one of you answer me A polypeptide chain number of amino acids. How many number of amino acid in A polypeptide chain and B polypeptide chain? How many? No, Allah. Think. Question mark. No question mark nowadays. Direct answer only. How many amino acids in A polypeptide? How many amino acid in B polypeptide? Any one of you? Hafiz, Sania, where are others? Answer me, Albert. Next. Shadi, what is the amino acid number in A? Is it 21 or 20 or 31 or 30? Anyone? No, you don't know the answer of this question? How many amino acids? Here A polypeptide chain is 21 amino acid and B polypeptide chain 30 amino acid, C polypeptide chain 33 amino acid. 21, 30 and 33 amino acids. Got my point? Done. Next question. So we can write option is C. We will have a question on this one. We will cross check. Now, a probe question. Everyone carefully get probe question. If you NCERT, open the NCERT and see. Probe is what? It is single stranded RNA or DNA which is complementary to the DNA. Or it is a DNA, single strand DNA or RNA. Look at here what is given. A probe which is a molecular basis to locate the homologous sequence of a mixture of DNA or RNA. With the probe, we can identify the particular sequence of a particular, what the sequence of DNA or RNA. Remember, if you have a NCERT, look at there, what is NCERT? Probe will not complement to the mutant gene. That's why the mutant probe will not be seen in the photo film given better in the NCERT. In the photo film, because the probe is not complement to the mutant gene. Remember all of you. So whenever you are adding the probe, it will be complement to a particular DNA or RNA. So it is a DNA and also RNA, single stranded DNA and RNA, radioactive one. So we can write what? It is single stranded RNA. Done. It's correct. It is a single stranded DNA. It is also correct. Either single stranded RNA or DNA. So I am going with option C. What about others? Very good. Absolutely correct. Next question. Very simple question. Which of the following commonly use a retrovirus? Vector for what? Lymphocytes. I'm sorry. That what is the lymphocyte? Commonly used for the vector to introduce DNA fragment into lymphocyte. Lymphocytes are the human cells. So human cells that commonly we use what? Retroviruses. So answer is option C. This mechanism we have seen in ADA enzyme therapy. So the lymphocyte, immature lymphocytes, with the help of retrovirus, we are administrating the desirable gene. Right? Are you with me? Next question. Question number 19. All of you pay attention here. Slowly I am explaining this one. Read the following statement. Fine. E. coli is a bioweapon How many of you correct? E. coli is a bioweapon. We are using E. coli for the different purpose, like a host. In genetic engineering, we are using the E. coli as a host. Is it true or not? Is it true or not? E. coli is a bioweapon is given. Fine. Okay. Agarobacterium tumefacium is used to as a vector for the introducing nematodal specific gene in the tobacco plant. Absolutely correct. B statement is correct. B statement, the second statement is absolutely correct. Agarobacterium tumefacium plasmid in the plant cell, right? Vector. The first ever transgenic cow is Dolly. Dolly is cow or sheep? Dolly is a cow or sheep? It is a sheep, it is not a cow. And moreover, it is not a transgenic, it is a cloned. It is not a transgenic, it is a cloned sheep. You got my point? So this statement is not correct. So I'm writing here, it's not correct. Alpha 1 antitrypsin is used to treat emphysema. Yes. Statement correct. Now, what is the right answer? What is the right answer? 1, 2, and 4. 1, 2, and 4. Option C. Option C. Am I correct? Come on, answer me. Am I correct? Right answer is option C. Are you there? Shall we go to the next question? Done. Next question. Look at here, the question number 20. How was insulin obtained before the advent of rDNA technology? What were the problems encountered? 2-mark question. I told you not only MCQs, I can also cover 2-mark question. What is the answer? Sir, simple explanation. Earlier, before genetic engineering introduced, the peoples are used to take the insulin from the cattle, pancreatic of cattle and pigs. But that non-human insulin is started what? Allergy. It started to show the allergy, so that the peoples are stopping that one, and genetically engineering insulin, human insulin is first time human protein or human hormone insulin is started to generate in rDNA technology with the help of Escherichia coli bacteria. Eli Lilly company, we have seen this one. No allergies. No, what they call side effect. This is a direct human insulin only. Direct human insulin. It is possible. We learn in molecular base of inheritance. Human insulin is possible to obtain from the bacteria because genetic code is universal. Whatever the sequence of mRNA codon in case of us, same thing in case of what they call the bacteria also. But you have to be very careful. You have to introduce cDNA into where? Bacteria. If you directly take the DNA and introducing into the bacteria, because in the bacteria splicing process is not there. So DNA to mRNA, mRNA to again DNA, and finally you have to ligate with the plasmid. So we can write what I explained you this answer. Simple answer. So 2-mark, this type of questions can come very simple. Next question. What is this? What is the answer of this question? Everyone aware about this one, I think so. So far, how many exams you attended in school? Pre-board 1, pre-board 2, half-yearly examination. This question, I think it must be there, I think so. So what is the right answer? What is this insulin? Select the correct set of names of A, B, C, D. What is A? What is this A here? Is a pro-insulin or direct mature insulin? What is this pro-insulin or mature insulin? Sir, it is pro-insulin. A is pro-insulin, then pro-insulin. So what is this B? A polypeptide chain. What is this C? This is A polypeptide chain. Polypeptide B, polypeptide C. Look at the C. C fellow gone out. Am I clear? C fellow gone out because the C, when removal of C A between the AB polypeptide chain, so disulfide bond formation, they become a matured functional insulin. So based on this information, what is the right answer? What is the right answer? Oh, very good. Absolutely correct. So pro-insulin is A. Next B, A peptide, B peptide, C free cDNA. So option B. This question might come for 3 mark, directly come for 5 mark also. Different higher order thinking question, different type of questions can come based on the same topic. So everyone keep in mind. 22nd question. Look at here. 22nd question. I told you this one. Any one of you pick up the right answer? Any one of you pick up the Hadia? Right answer. Ritika, fast. Shadi, Ganga, Allah. Next, who is this? Sania. Very fast. The answer of the how many amino acid in C polypeptide chain is given. Absolutely. Very good. The polypeptide chain is removed during the maturation of proinsulin A. Proinsulin polypeptide C is removing 33 amino acid. Remember all of you, A polypeptide chain 21 amino acid. That's why it is given short DNA. Look, come back, go back to the previous. Look at here, short. I'm sorry, not short polypeptide chain. Look at that short polypeptide chain because 21 amino acids are there, and the B1 is little big. 30 amino acids are there. 30 amino acid. I have a question, sir. A polypeptide chain and B polypeptide chain for the synthesis, different genes are there, or only one gene will give A polypeptide and B polypeptide both? Any one of you answer me? Pavitra, can you answer me? Sir, A polypeptide and B polypeptide chains are transcribing and translating from the same gene, or for the A polypeptide chains different gene, B polypeptide different gene? Yes, absolutely. Absolutely. What's the right answer? Good. What about others? Sir, it is necessary to know that amino acid. Yes. Because, Albert, sometimes this type of questions can repeat. So you don't know the number of amino acids, you will lose the MCQ. You don't know the amino acid number in AB polypeptide chain. How can you answer this type of question? So it is necessary to know amino acid number also. So A polypeptide chain is different, B polypeptide chain also different, particular gene is there, that gene undergo transcription translation to give a particular amino polypeptide chain. Done. So I need what is the 22nd answer? Option C. 23rd one. Any one of you? 23rd. A patient has a defective gene for the enzyme adenosine deaminase. He and she lack functional cells of, therefore fails to fight the infection pathogen. The cells are. I told you in the last class. One more time. Look at here. This is bone marrow. All of you look at here. This is bone marrow. Now in this bone marrow there are specific what they call stem cells are there. This is bone marrow. Now this bone marrow, this stem cells differentiate to give B cells and also B lymphocyte cells and T lymphocyte cells. But B T lymphocyte cells in whatever the DNA is there, in this DNA the gene ADA gene deleted, no gene. So no gene, no transcription, no translation, no adenosine deaminase enzyme, no ADA enzyme. When ADA enzyme lack, B T cells are immature. Remember all of this. Immature B cells not going to fight against the pathogens. Got my point? So where is the genes located? The gene located is B cell, T cell. So answer is option D. Am I correct? Oh, very good. Absolutely correct. You all are very fast. Then very good. Next question, 24th one. 24th question. Chalo. How fast you are? Come on, answer me. Which of the following transgenic human protein produced development of via rDNA technology used to treat emphysema, lung disease? Very simple, sir. Alpha antitrypsin. Am I correct? Very good. Alpha antitrypsin. Cry2AC protein sensitive to where? Cry2 protein is very affected on what type of insects? Answer me. We discussed this one. Hafiz, Sana, come on, any one of you, Jade, come on. Cry2AC protein acts on stem borer or cotton bollworm. Answer. Very fast. Hadia, come on, beta. Come on. Ritika, sir, it is cotton bollworm. Cotton bollworm or stem borers? Yes, cotton ball. I told you cotton bollworm plus Cry2AC and Cry1AB. Remember, right? Next question. Consumption of which one of the following food can prevent the kind of night blindness? This we discuss. Consumption of what? Consumption of what can prevent that? Night blindness. Anyone. Very simple. What is that? Golden rice, bio-fortified, vitamin A enriched rice. One of the transgenic crop which can prevent the night blindness, this eye problem, this ophthalmic problems, right? This is enriched of vitamin A. Next question number 26. Bt toxin protein crystals prevent in bacteria Bacillus thuringiensis. Do not kill. Bacillus thuringiensis is a bacteria. Cry protein synthesis, cry gene is there, which undergo transcription and translation to give a cry protein, which is very sensitive on this lepidopteran, dipteran, coleopteran, beetles, mosquitoes. We have done this one. But why this cry protein is not hurting, not affecting, not killing the bacteria? Very good, absolutely done. Because for the activation of this cry protein, alkaline pH required. Where is the alkaline pH in bacteria? There is no alkaline pH. So if there is no alkaline pH, remain it what they call inactive. What is inactive? Bt toxin is inactive in the bacteria. But once it get activated in the gut of insects and caterpillar, khatam, it will be killed. Done. So answer, what is the answer now? Toxin occurs in the inactive prototoxin. Option C. Biotechnology chapter, application chapter is very easy if you compare with the biotechnology process. In biotechnology process, lot of things are we need to understand. PBR322, PCR, that, this one. But here only RNAi mechanism, gene therapy, insulin, transgenic animals, whatever the functions of transgenic animals, bioweapon, biopiracy. Only these are the topic. I think if I am not wrong, 2 hours enough to revise the entire chapter. Biotechnology application chapter, 2 hours enough to complete revise. Done. Next question. The first human hormone. Any one of you? First human hormone produced by rDNA technology. First human hormone. Anyone? Very good. Absolutely correct. Insulin. If you able to recall in the NCERT, it's given therapeutics. Therapeutics are 30 therapeutics so far. I've been isolated, discovered, isolated, prepared by rDNA technology. How many in India? 30 worldwide therapeutic have been isolated from the rDNA technology. How many? Very good. Absolutely good. 12 in India. How many in marketing in India? 12 marketing in India. So we can write insulin. Option A. Estrogen, thyroxine, progesterone hormones are given. The first rDNA technology human insulin hormone. Next question. Transgenic animals are those which have foreign DNA or foreign gene. Carefully read this question. What is the meaning of this one? You created a transgenic mouse. You created a transgenic pig. That means you introduce a human gene into that animal or any gene into that one. So that gene is specific one location only or all over the body? Shadi, done. Very good. It is specific to a particular organ or all over the body? I need answer. Now look at here. So DNA in all of their cells, protein in all of their cells, RNA in all of their cells, RNA in some of the cells. Is it some of the cells or whole body? I need answer. Sir, whole body, yes. How come it's belong to a particular area? No, right? It will be the whole body, but functional to a particular organ. Remember, function is a particular organ, remember, right? So I can write the answer is option A. Done. Very good. Absolutely correct. Next question number 29. The genetic defect adenosine deaminase deficiency may be cured permanently by. They're asking enzyme replacement therapy, right? No, sir. No enzyme replacement therapy. Bone marrow replacement therapy, right? No, sir. Gene therapy, right? Yes, but early embryonic stage you should identify, you should detect the disease, you should introduce into the bone marrow cells of early embryonic stage, because the early embryonic cells are stem cells. So introducing bone marrow cells, provide ADA into cells at early embryonic stage. Option B. How many of you correct? How many of you correct? Absolutely. Next question. ELISA used to detect virus where the key agent is. Key reagent in ELISA. It's not there in the NCERT, but I told you this one before. Jade, answer me. Samuel, yes, Hafiz, correct, very good. And then next, Shadi, right, very good. Ritika, what is the answer of this one? I told you the alkaline phosphatase. The reagent is alkaline phosphatase, providing a particular color. Next questions. Can you do this? BT cotton variety that was developed by the introduction of toxins gene Bacillus thuringiensis resistant to. Sir, option B, very good. Next Samuel, next Sana, you should answer, come on. Sir, it is a fungal disease. Bacillus thuringiensis is not fungal disease. Insect pest, plant nematoda, insect predator. I am going with D, insect predator. Is it correct? Is it insect predator or insect pest? Answer. Sir, it is insect pest, right? Not insect predator. Insect predator we discuss, insect predator we discuss in microbes in human welfare, bio fertilizers, biocontroller agent we discuss there. But here it's not. It's what is that? Insect pest. So I'm going, answer is option B. Done. Very good. Absolutely correct. Next question. Wait, all of you. Last 5 minutes, carefully listen. So far MCQs we discuss, 2 more questions, accession reason we discuss, 3 mark questions 2 and 2 mark question 1. Let me take the first one. What is gene therapy? If you read NCERT, clearly it's given, if a mutant gene or problematic gene, it is a process in which the insertion of a desirable gene in place of what? A mutant gene or a problematic gene. Gene therapy is that technique to insert a proper gene into the defected area with the help of a vectors. So this gene therapy can diagnose this gene therapy can heal or cure the particular genetic disorders such as SCID, such as cystic fibrosis. Now what they given here, illustrate using the example of adenosine deaminase deficiency. Adenosine deaminase deficiency is caused by the deletion of ADA gene. Through gene therapy, we are inserting this bone marrow gene into the lymphocytes of B and T lymphocytes and make them functional. But this process will not give a permanent curing, because the lymphocyte cells are not immortal one. They are mortal one. They will die. Within 15 days or 1 month it will die. So to get a permanent cure, this bone marrow cells or gene therapy have done in bone marrow cells at early embryonic stage. Gene therapy questions, a lot of questions are going to come, my dear students. So be careful. This is a very simple question. Done. Next, does our blood have protease and also nucleases? Answer me. Does our body has protease enzyme and nuclease enzyme? I will be silent. Any one of you? Just your answer is say yes or no. Sir, our body protease. Sir, wait. Protease enzyme function is, let me write here. Let me give a hint. Protease enzyme function is digestion of or degrading of proteins. Next, nuclease enzyme is also degrading of DNA and RNA like enzyme. Tell me, what they're asking, does our blood, sir, in our blood, a lot of proteins are there. Plasma protein, immunoglobulin, prothrombin, globulins and albumin proteins are there. Serum proteins are there. If protease enzyme is present in our blood, it will be digest all this one, it will be degrade all this one. So is it happening in our body? No, simple. Remember, our blood does not have protease enzyme, but protease enzyme secreted in our alimentary canal in different area. Am I clear? But blood does not have nuclease enzymes also. This type of questions can repeat. This type of question can come. Our blood does not have proteases and nucleases. If it present in our blood, they will degrade our blood protein, serum proteins. Are you with me? Are you there? Done. Next question. This question, carefully look at here. What is the question is given? Differentiate between diagnostics and therapeutics. Give one example for the each category they are asking. Careful listen here. 2 minutes. Everyone careful listen here. Diagnosis means it is a technique to identify the disease. Diagnostics are the techniques to identify the disease such as ELISA, PCR, like this one. It is called a diagnostic one. What is the therapeutics one? Therapeutics one are the one which inject in the proteins or which inject in the hormones like. So therapeutics are like rDNA insulin production, rDNA estrogen production, or different type of protein production. That's called the therapeutics. So remember student, if this type of questions come, you should carefully answer, otherwise we will not get 100 out of 100. We are taking these classes to get 100 out of 100 in your final examination. So if you don't know this type of small questions, it will be loss. So therapeutics means rDNA technology, whatever that vaccinations also therapeutics will come. rDNA technology, insulin also therapeutic will come. FSH injections like LH injections like therapeutics will come. So what are diagnostics one? That ELISA, PCR, these all are detection of diseases called a diagnosis. Am I clear till here? Everything done? Am I clear? Last question I am going to, this is a very important, look at here, 3 mark question. Diagrammatically represent the experiment steps in cloning and expressing human gene. The gene for growth hormone into the bacterium E. coli. Wait a moment. After I finishing this question, you can move. Listen to me very carefully. Diagrammatic representation they are asking. Look at here carefully. What I have done, I took a plasmid of E. coli bacteria. Look at here the E. coli bacteria plasmid, and this is a human cell and gene I extracted. Isolation of gene, and after isolation we learn the biotechnology steps. Fragmentation. Look at here. After that, gene extraction, treatment with the restriction enzyme, fragmentation, specific gene of growth hormone I took, and with the help of restriction endonuclease enzyme I am also cleaving the plasmid. So this plasmid and this gene, that means our desirable gene, I am combining to form rDNA. rDNA again I am transforming into where? Host cell, bacteria, and bioreactor method. Lot of bacteria are producing. All bacteria are expressing mRNA and also protein. And in the bioreactor, I am increasing the number of this bacteria and finally obtaining my desirable product. The product is growth hormone. Growth hormone with what? Tell me. rDNA technology. You have to drop this diagrammatical. This type of question can come for 5 marks. Are you with me? So this is today's class, biotechnology application. So thank you all of you attending this all sessions, and moreover I wanted to inform you something. Today is last biology live YouTube class. We are going to give a break, and after that, before bio exam, before chemistry exam, before mathematics exam, there will be a live a rapid revision classes are going. My dear student, that will be 100% helpful for you. Only MCQ, not only MCQ, 2, 3, 5 mark questions also we are discussing the rapid revision. Am I clear? Thanks for attending all my classes, all the brilliant YouTube classes, and these classes I hope these classes help you to brush up your complete concept, and these classes will help you to get 100 out of 100. Thank you very much. And whoever not attended, whoever your close friends, share this video. Am I clear? Ask them to watch and ask them to also increase their knowledge or content. And all the very best for your final examinations. My small suggestion is, whatever the chapters you're feeling difficulty, spend more time on that topic, that subject, that chapter. Am I clear? All the very best. Thank you very much. Good night, all of you.
Brilliant Qatar, your trusted coaching partner for IIT, JEE, NEET, Science and Commerce tuitions with 10 years of excellence in quality training. Brilliant Qatar.